The possibility that life has not originated exclusively on Earth has been part of the scientific debate for a long time. Researchers have argued that the chemical ingredients necessary for biology could have come from space, transported by meteorites or comets. In this context, some scientists have explored the hypothesis that microscopic organisms can travel between planets within fragments of rock expelled by large impacts. If you're interested in the fascinating world of astrobiology and the mysteries of the universe, don't forget to subscribe to our channel and like this video. Here we explore themes that could change our understanding of life in the cosmos and beyond. This scenario, known as lithopanspermia, raises the fundamental question: can microorganisms survive the extreme conditions of an impact? A new study published in PNAS Nexus addresses this problem through experiments designed to recreate the pressures that would occur when ejecting rocks from Mars. Researchers combine impact physics techniques with biological analysis to explore the limits of microscopic life. The impacts of asteroids have been influential processes in the history of the solar system. These events have shaped the surfaces of planets and moons and have modified their environmental and chemical conditions. Impact is one of the dominant processes in solar system, responsible for the formation and alteration of many planetary bodies. When a large object hits the surface of a planet, it generates extremely high pressures for brief intervals. These conditions can destroy geological structures, but also expel fragments of rock at sufficient speeds to escape gravity. On Earth, meteorites have been found from Mars, which proves that this transfer of material between worlds is real. If Martian rocks can reach our planet, one wonders whether the microorganisms trapped inside them could survive the entire process. This scenario is the nucleus of the hypothesis of lithopanspermia, which poses the natural dispersion of life through fragments of rock and expelled by giant impacts. To investigate this issue, the researchers designed an experiment that recreates pressures comparable to those generated during an impact. They used a metal plate assembly where biological samples were placed. A gas-driven projectile hit the structure, generating an intense pressure pulse for a fraction of a second. The experiment subjected bacteria to pressures of between 1 and 3 gigapascals, levels that can occur when a rock is expelled from Mars. At 1.4 gigapascal, approximately 95% of the bacteria survived. At 2.4 gigapascals, around 60% remained viable. This shows that the extremophile D. radiogerns has a remarkable resistance. The study not only measured the survival of the bacteria, but also analyzed their reaction at the molecular level. It was found that after the impacts, genes related to DNA repair were activated, indicating that the bacteria not only survived, but also repaired the damage. These results do not demonstrate that terrestrial life comes from Mars, but they provide valuable information to the scientific debate. If some microorganisms can survive the pressures generated during the expulsion of rocks, at least one stage of the interplanetary journey could be feasible. The finding has implications for astrobiology and the design of space missions. This work suggests that life could be more resilient than thought. The natural transfer of life between planets may not be as unlikely as it seemed.